I'm the acting director of the Human Brain Collection Corps, which is a brain bank in the intramural research program of the NIMH. So our mission is to support and conduct research on brain and behavior with the goal of reducing the burden of mental illness. And our more restricted goal is to identify molecular mechanisms of psychiatric disorders. And post-mortem research in this realm is critical because psychiatric illness is difficult to model, and the tissue is difficult to access in life. So there are two arms, two main functions to the Human Brain Collection Corps. One is brain acquisition, and the other one is distribution of biospecimens. And I will describe both of these. So we collect brains from, based on referrals from the Office of the Medical Examiners in DC, in Northern Virginia, and in Central Virginia. They refer the cases to us. We call the family or next of kin, and we ask them whether they are willing to donate the brain for research purposes. If they agree, then we record their consent and we relay the family's decision to the medical examiner. And the medical examiner extracts the brain for us, and we either go and pick it up or we have it shipped to us. When we receive the brain, we test for HIV, for hepatitis B and C, and then we are currently testing since this summer for COVID, both with PCR and with serology. We cut the brain in coronal sections using a mold that is helpful in making those cuts more consistent from brain to brain. And I'm sorry, I have a thing superimposed. And then we flash freeze the tissue, and we barcode and all the samples, and we track everything in a database. We have more than 75,000 specimens barcoded at this point. So we do a lot of work to characterize the tissue that we collect, starting with the neuropathological diagnosis that is done now in a very in a way that is consistent with all the more current staging systems for Alzheimer's disease and for neurodegenerative disorders. We measure the pH, and especially the RNA integrity number is important to us because we consider this as the more one of the most important measures of tissue degradation. We then do forensic toxicology on hundreds of compounds on all the brains, all the blood that we receive together with the brain. And we genotype all cases. And currently we are in the process of doing whole genome sequencing on all cases. We interview the next of kin, and we collect the report from the medical examiner. We calculate postmortem interval. We collect medical records, as many as we can, and especially psychiatric records. Then we put together a clinical summary, and then two psychiatrists review all this information and come up with a consensus diagnosis, not only for psychiatric disorders, but also for medical conditions. We have some inclusion criteria, so we are looking for cases with a presence of mental illness or substance abuse or suicide, and also for non-psychiatric comparison cases who have not had a psychiatric disorder or a neurological disorder in their lifetime. We exclude if the brain is too decomposed, if the subject was on a ventilator for more than 12 hours, if they died of sepsis, if they had infections affecting the brain, if they had hepatitis C or closed head injury or trauma, or lesions of the brain that have destroyed the tissue, history, long-standing history of a seizure disorder, of multiple sclerosis, and we also exclude if we find signs of dementia or the degenerative disease at the neuropathological stage. Our entire collection consists of about 1,100 cases. The RNA integrity number is quite high, so it's around 8 on average. The post-mortem interval, so RNA integrity goes from 0, that is very poor quality, very degraded RNA, to 10, which is the best quality. The post-mortem interval is around 30 hours, and the age of the collection is between 30 and 60, so relatively young age for other, compared to other brain banks. And one of the advantages of our bank is that we have quite a few subjects in the very young individuals. The collection is primarily male, less so female. And you can see here the number of cases in the various psychiatric disorders. So we have a large set of controls, but also many cases with schizophrenia, bipolar disorder, major depression, and other less represented disorders. There's a lot of comorbidity in psychiatric diagnoses and substance abuse diagnosis especially. So this is a number of subjects. The blue bars are what we call a primary diagnosis, so the most important diagnosis that that person is carrying. And the orange bar are if the substance use disorder, for example, is present in addition to a primary diagnosis, so, or in addition to another psychiatric disorder. So you can see that the group with the biggest difference between all diagnosis and primary diagnosis only is the substance use diagnoses, but also other primary diagnoses of anxiety disorders, for example, PTSD, or attention deficit disorder, have high comorbidity with other disorders. We use currently the DSM-5 criteria to diagnose. We have retained the axis 1, 2, and 3 subdivisions because we think those are useful. And so yeah, that's the general. We have a lot of information that is readily available about these samples. That includes demographic information, manner and cause of death, neuropathological diagnosis, psychiatric diagnosis, history of smoking, education, socioeconomic status, height, weight, and extensive forensic toxicology. And now we're collecting also data on place of birth or trauma exposure. And we're in the process of adding DSM-5 criteria for all the cases. We characterize the cases genetically. So we've used genotyping, microarray technology up to 5 million chips on all cases, and we have a whole genome sequencing on 400 cases that are now, and we are now in the process of doing whole genome sequencing on another 600. All the inventory is accessible through the Neurobiobank website. From the tissue distribution standpoint, there is a request process, so the investigator will send us a request. We have an extensive consultation with the investigator to make sure that our resources are best utilized to satisfy that request and to solve the specific scientific question that is being asked. Once we are satisfied with that project process, then we pass it on to an oversight committee and if the oversight committee approves the request, then we put a material transfer agreement in place, and then we carry out the request. And the timing here depends on how busy we are satisfying other requests and on the complexity of the request itself. We provide many types of biospecimens. As you saw earlier, we freeze these slabs and then we can dissect from them with a dental drill to create frozen blocks of tissue. And those can be pulverized for certain applications. We have sections on slides. We have a large collection still remaining of slides from the dorsolateral cortex of 32 patients with schizophrenia and 63 comparison subjects. These we are trying to give away because they occupy a lot of space, and we are starting to have space issues. And but we can also cut specific, we can create slides, sections on slides of specific brain regions for specific projects. We also have a few brains that are fixed in formalin, a single hemisphere. However, we can also always derive from the slabs fixed or fixed tissue. So we also collect blood. So we have blood for many of our subjects, and we have recently started collecting PAX gene tubes that preserve the genetic material more in an improved way, and we hope that those will be helpful to the requesters in the future. We like to extract DNA or RNA from, if the request is centered on these nucleic acids, we can do that for you. And this is best for us because we can retain a part of that material, and that can be used in the future to validate certain findings or to satisfy other requests. We have also created cDNA libraries for qPCR studies for different regions of the brain, and several in large numbers of cohorts of subjects, mostly including schizophrenia and controls, but we also have other cases in the dorsolateral prefrontal cortex and the hippocampus, for example. We have also conducted RNA sequencing studies in several brain regions, and I will mention those a little later. And we also have some fibroblasts derived from postmortem dura of 450 subjects. We also can extract nuclei, and now we can do library preparation with 10x Genomics methodology for single-cell sequencing. So our main goal is to share the resource and the data as widely as possible. We started by posting on dbGaP our microarray RNA expression studies of the dorsolateral prefrontal cortex, the hippocampus, and the dura. We have then collaborated with the Common Mind Consortium and PsychEncode to post data on RNA sequencing, chromatin immunoprecipitation sequencing for histone marks, and whole genome sequencing of the dorsolateral prefrontal cortex in a large group of non-psychiatric comparison subjects, schizophrenia cases, and bipolar disorder. And we have also made available data for RNA sequencing of the subgenial anterior cingulate in 200 subjects, including cases with major depression, and for the superior temporal gyrus and the dorsal anterior cingulate. And this was done in collaboration with the human genetics branch at NIMH. Now, our ultimate plan is for all genomic studies, whether conducted at NIMH or conducted by requesters outside, to publish this on the NIMH data archive. So the hope is that over time, this will become a very rich collection of data on the same subjects with different, with multiple methodologies. And this effort of data sharing has given origin to at least two well-published data sets. One is on scientific data and contains our RNA sequencing data from that we shared with the Common Mind Consortium, and the other one was published in Science in 2018, based from the PsychEncode group. And we hope these resources will be helpful to your research. And these are the people who have done all the work. It's a group of about 15 people, and we owe a debt of gratitude to the medical examiners, to our close collaborators within and outside of NIH, and to the families of the deceased. So that's about it. If you have any questions, I'm happy to answer them.
I actually have a comment to make, and I just want to say that I've been using some of the samples that Dr. Marenco was talking about. Those are the dorsolateral prefrontal cortex slides from schizophrenic patient and healthy control. So we have conducted an immunohistochemistry study looking for some axonal markers that are of interest for us. So the mechanism was, of being able to access the tissue was really, provided really an amazing opportunity. And this was one of the reasons I asked Dr. Marenco to provide us this presentation, because I think that it will be very useful for our group, for our cluster. So
I have, Cielo, I imagine that you have a lot of questions. Can I just a comment? about what Fernanda was talking about sharing the data. You mentioned at the end that the data are actually the standard analysis that you run basically are already public. It's just if a researcher needs the specific tissue that there is that two-step process that you discussed, Dr. Marenco. Am I right?
Yes. So you can request the data from NDA or from or with more limitations from the CMC consortium. So those are kind of public access requests where you have to, submit a plan basically, and there's some control on how the information is given out or to who. If you are doing a new study, then you can request to us, and we would evaluate the request and get back to you, as we did with Fernanda.
This is Samina Salim from University of Houston. Thank you for an excellent presentation, very useful and informative. I had a question about, so people who request these tissues, do they have to be funded from NIH, or that's not?
So we actually distribute tissue to the whole world, so it doesn't matter whether NIH is the source of funding, but we do ask whether you're funded to do the research. So we don't want to give a tissue to somebody who's not funded yet, and who might not be able to use it because they don't have the funds. We do provide letters of support for requests that for funding. So if you are writing a grant and you would want to use our tissues, you can ask us for a letter of support. We will evaluate the request. If the request is, if we can satisfy that request at the time that we receive it, then and the oversight committee agrees that it's a good scientific purpose, then we'll send you a letter of support, and then if the grant is funded, then we'll give you the tissue.
All right, thank you.
Any other comment or?
No, I'm sorry. I was actually a little late, but I just think this is a great opportunity. I have a brain collection. I don't know if all of you know, but it's mostly addiction, subjects. So it's good to always expand and look into other phenotypes.
What is the source of your brains? Medical examiner's offices?
Yes,
or
it's here in Houston. It's the medical examiner's office exclusively. We get them all from the coroner's office, and so I think that's why most of our here in Houston, the addiction crisis. So most of our subjects that we get are people that died of an overdose, and mostly opioid overdose is what we're getting right now.
So we have the same type of issue, as you saw, or maybe you were late, but there's a lot of comorbidity with substance use disorders.
Yeah.
However, our bank was formed from the collection that Joel Kleiman had initiated in the 1990s. So that is why we have such a large number of
Schizophrenia.
cases with schizophrenia and bipolar.
Yeah, that's really good. We don't, we do a detailed psychological autopsy and get all the, medical records and all that. And I think we have 2 schizophrenia cases. We have like over about 130 brains right now.
Mhm.
And yeah,
Yeah, but that's also my experience right now with getting brains from the medical examiners, that if you get 2 or 3 a year, that's good.
yeah. But it does take a village. I like your slide there. It's a lot of work. It is a lot of work.
So that's yeah, I think that's great. I think it's the idea again of this meeting was to hopefully spark interest in initiating a collaboration with your center, basically, so that we have a lot. I think there were I don't know, Cello, if you were already online, but what Dr. Marenco was talking about is, I think it's a new feature in which you can isolate actually single cells. You were on, Cello?
No, not yet, but no, that's
Right.
the latest thing in postmortem is to get
Yeah,
single
what was
cell.
Doctor, was it slide 12? If you can show it again one more time. Right, yes. So yeah, I thought that was very very innovative, and the possibility of extracting single cells, extract nuclei. So can you tell us just a little bit of what the status is on extracting nuclei? As Dr. Vaspas was saying, that's definitely the new frontiers, and we actually here in Houston at the Texas Medical Center are very, very well equipped with tools to assess single cell features. So
we've done one initial study where we took 8 comparison subjects and who had no psychiatric or neurological disorder, and did the single cell sequencing of the dorsolateral prefrontal cortex and the subgenuine anterior cingulate, 2 areas that are important for psychiatric disorders. And we actually went all the way through to validate with RNA-scope the findings, the different cell populations. And I think we're putting together a manuscript now, but we have results that are fairly consistent with what has been emerging from the literature. And more recently, we did 24 patients with schizophrenia and 24 controls in the dorsolateral prefrontal cortex, and we're still waiting for the sequencing results of those. And even more recently, we acquired the machine that allows us to do the library preps. So, and we're starting to work on that now.
now the 24 that you're talking about with schizophrenia and the controls are the dorsolateral prefrontal cortex. Are those the same patients that we have studied on, that are mounted on slides right now? Is there a way basically to combine, or to enrich any of the data on the slides with something that is new that we weren't aware of a couple of years ago when we started?
Potentially, I am not sure. I would have to see the list of your subjects, and but you could already go to look in NDA.
Mm-hmm.
If there are any data, any sequencing data associated with those, and then I can tell you whether any of those ended up in the in the single cell stuff.
Yeah, I think maybe the easiest way for us will be to send you the manuscript. At least, I mean, of course, we can also have an, you know, a list, a summary list of, you know, the specific code of each patient. That's okay, but maybe that would be helpful. We had to spend a lot of time to find a statistician to make a proper comparison with all the imaging features that we analyzed, and so we have completed that part, and so now we can basically work toward the final analysis of, well, not the you know, to finalize the manuscript. I think that would be very useful if we have any additional possible data that we can use.
Sure.
Thank you.
Oh, can I ask a question about the, I see you have fibroblasts that were derived from the dura. So I assume that the idea is that then you can make like iPS cells, or what are you
Yes.
guys?
Yes, that is in theory possible. I don't know. This was a resource that was left over from Joel Kleiman, and we have never accessed it so far. So I don't know how usable it is.
Okay. So yeah, okay. That's interesting.
So you would have to, if you're interested, you would have to give it a try yourself, basically. So.
Okay. Okay. Yeah. But you don't, those 450 subjects, you don't know anything about their phenotype?
Well, mostly patients with schizophrenia controls, and then
Okay.
maybe
Oh, that's
a
okay.
few bipolar disorder cases.
Okay.
So our plan is to, you know, that was, I guess we said that at the very beginning, we're going to share this presentation with other steering committee members who weren't able to make it today. Usually we have our monthly meeting the second, I think it's, yeah, the second Wednesday. So it would have been last Wednesday. So for some people, today wasn't the, you know, the exact schedule. So, but anyway, I think, again, I think it's really, really great. So do we have any other questions or comments? So I guess if we don't, you know, we can let you go.
Okay.
Dr. Marenco, thank you so much. Again, we will distribute your talk and obviously the information, your contact information to all the members. And then we'll go from there.
Okay, great. Thank you very much.
Thank you so much. Thank you.
Okay. Bye-bye.